So, good evening. July 10th, we have concluded a summer internship in Limnetic Microbiology. It's a high time I thought we will do a, what to say, a highlights video on summer internship in Limnetic Microbiology to explore what we did and what is the impact of this program. So we started this internship in June 10th, inviting students from various parts of India, especially from National Institute of Technology, Vellore Institute of Technology, and Crescent University, Rajalakshmi Engineering College, St. Joseph College, and Krishna Jayanti University. So we initiated the internship with the lab tours and a few fun activity to familiarize the interns with basic microbial techniques. We had a competition and then we prepared for a big mission of sampling the Dudahabi Lake. Actually, a funny story. We initially targeted Kalkeri for some reason we couldn't sample it. So we adopted Dudahabi, and later we realized we are very glad that we adopted Dudahabi because it is relatively pristine lake. On June 16th, we went for sampling. Each and every intern, there are more than 39 interns precisely, so including volunteers. They have their own sampling plan, and we practiced how to sample in the lab before we reached the Dodahabi Lake. Yes, the lake had various phytoplanktons, diatoms. So we microscopically documented them, and then we started our spread plate analysis. So we inoculated all 39 samples into nutrient agar and various other media, where the intention is to study the bacterial morphotypes, fungal morphotypes, and also we selectively look for potential pathogens. So we initiated with the abundance calculation with the serial dilution. We try to calculate or count plates between 30 and 300 CFUs. So we realized by this time that we cannot work just between 9 a.m. and 5 p.m. So we extended to 8 p.m. Literally, we are working between 9 a.m. and 8 p.m. during the internship. So here is a master plate or mother plate. Based on the colony morphological features, we decided how to go about it. And the good news is there is no salmonella, or another way of saying there is no black colonies in XLD agar, which indicate the lake is free of salmonella. So the interns were trained in pure culturing from the mother plate, how to identify the unique morphotypes, how to streak them and pure culture them, and how to do last quarter metrics. Similarly, for fungi, how to isolate them and purify them for pure culturing. And yeast, we also have a yeast species. So we trained interns on how to differentiate bacteria from yeast. And yes, we targeted microscopic algae to microalgae. We got two species. One is chlorella, and we are still working with the other, and the interns have purified them. We studied macromorphological features of individual colony morphology, and then we moved into micromorphological features through gram staining. Out of the entire spectrum of organism collected, dominant was gram positive, gram negative rods. As it is an aquatic ecosystem, we have 144 strains, which is actually positive for catalase. We have 146 strains positive for oxidase. Endospores, we have 23 strains. And we subjected all the strains to biochemical tests like indole methyl red Vogus citrate for gram-negative bacteria and motility for both gram-negative and positive. For positive, especially, we went for endospore staining and we used amylase production, that is starch hydrolysis, as a key character to differentiate Bacillus from Clostridia. We in Krishna Jayanti University Department of Life Sciences, we use spectral signatures to discriminate bacteria. So we trained interns in taking spectral signatures of their broth between 200 nanometer to 1100 nanometer using UV spectrophotometer. And most of the spectra is within 200 range. And to look beyond what is there, beyond 1100 nanometer, we adopted Fourier transform infrared spectroscopy, FTIR. We also created spectral signatures for FTIR of the isolates. So all gram positives are actually screened for amylase production through starch hydrolysis, and we have a TSA agar for our 200 plus strains that is screened for it. So these are the actual selective media we used, Xyl, XLD for Salmonella and Shigella, and we used starch casein agar. So XLD and starch casein, we couldn't find any organism, but in citramide agar, we do got Pseudomonas aeruginosa, and the plates were full of EFB. So there is a fecal contamination in the Dudahabi lake, but not as strong as other lakes. So we have cyanobacteria too in blue green. We used for cyanobacteria, but we end up getting microalgae. We screened all 200 plus bacterial strains for these antibiotic sensitivity assay. We are very much surprised to see uh, more than 54 strains are actually resistant for kanamycin, 42 are resistant for azithromycin. So like this, even for amoxicillin, more than 10 are resistant. So the lake is harboring diversity of antibiotic resistant microorganisms. So after these applications, we trained the interns in molecular characterization where interns actually isolated DNA plasmid. They also learn how to use STS page in profiling the bacterial protein profile for discrimination. So around 111 bacteria were out of 200 and plus strains. 111 strains were actually selected for sequencing based on comparison of all these macromorphological, micromorphological, and biochemical and molecular characteristics. We have also for sequencing, and we have produced around 111 6NS rRNA gene sequences for bacteria belonging to 52 species. All intents have submitted at least published one 6NS rRNA data in GenBank. Thereby, this internship has produced around 111 sequences. Interns were not just stopped there. They're actually trained in blast analysis, how to use the sequence to find the species, how to interpret the blast analysis using phylogenetic tree construction. All those bioinformatics analyses were actually trained for the interns, and they are making their own technical report now. And these 183 plus species that strains we have recovered are actually preserved in glycerol. It is deposited in Christogenthi Microbial Culture Collection Center. And these strains are, of course, available for further academic and research purposes. Right now, 111 sequences were published in GenBank. And for fungi, there were around 50 species of molds and yeast where ITS region sequencing is been happening, and soon it will be available. So here is a quick glimpse of what we did through summer internship. These are the published sequences. You can see Kristu Jayanti strain ID. We have a GenBank accession number, the name of the species we have recovered through summer internship, and here is the GenBank link. We are very much happy to see various organisms, various interesting organisms. In fact, Aromonas caviae, which is most of fish pathogens. We have Commonomonas testosterone, which can use testosterone as a carbon source. We have Lephonia aquatica, and we have Shivanella putrefians, all most interesting organisms. So we did not stop here. We want to see how to use these bacterial strains. So we went for bioprospecting, in which we checked actually, can we use organic dyes to stain these microbial cells? Can we use these organisms for producing nanoparticles? Why not we try to isolate phages from these organisms? Hydrocarbon degradation, nanoparticle synthesis, bacterial antibiotic production, fungal antibiotic production. So we actually screened through cross-streak assay to check for bacterial antibiotic production. Here you can see KJU 230, it's actually inhibiting KJU 0222. So they are not growing nearby that. So these are potential candidates for bacterial antibiotics. We also went for pigment producing bacteria. We had a lot of pigment producing green, yellow, pink, red. So we try to extract pigment to see whether we can use it for a food colorant or what is the toxicity status. So the work is still in progress. We synthesized a silver nanoparticle using a bacterial isolate isolated from the lake. And using FTIR, we confirmed the presence of silver nanoparticle. We also studied heavy metal and hydrocarbon degrading bacteria from the lake sample. We prepare lake, plates with various concentration of heavy metal and hydrocarbons, and we have screened their bacteria for this activity. We adopted submerged fermentation for fungal antibiotic production. There were very few candidates for which can inhibit the potential bacterial pathogens here. So we are still working on that. So why not I use a beetroot juice to stain a microbial cell or butterfly pea extract to stain a microbial cell. Intern were also trained in using these organic dyes to stain it. So the work is still there, and all the strains we produced through the summer internship has been deposited in Kristi Jayanti Culture Collection Center, and we got anyone who is interested in exploring these strains can easily access through this website and order the strain. We also had an award called Emerging Microbial Limnologist awarded based on their active participation every day and quality of documentation, documentation and result discussion, and also innovative approach to maximize whatever they are doing. So we have awarded this to an external intern from VIT. And we also brought in our alumni who is running microbial startups to discuss on what are do's and don'ts of doing a microbiology startup. We also had our alumni training our students in various specific skills like bacterial protein profiling using STS page. Yes, when you have students from all over India, we found an innovative way to bridge them. Doesn't matter from which country of India they are from. We made sure they had a good time. It's not about just incubating ourselves completely in the lab. It's all about having a vibrant time so that we will have a good bond. The interns will had a actually they had a good bond and they become very close friends due time. And for valedictory, we brought CEO of Genofi. We gave the report of internship and the outcomes of the internship. He was so much impressed, and we had a. He advised us, or he has given various productive suggestion how to take this internship further, how to bioprospect these organisms that we have produced during the internship. So there could be a summer internship or another winter internship during December, or there could be a workshop to explore bioprospecting aspects, or there could be an online internship too in near future to explore to apply AI in species detection, as we have various data now, plate pictures, biochemical tube pictures. Now it's we also know the species name through molecular data, that is 6ns RNA gene sequencing. Now we can integrate all this data to form a to build AI system which can actually identify bacteria from their images. So such a work is still happening. So that's how we did this 30-day summer internship. Thank you. Keep watching Unknowns. We'll update you shortly.